Hi, my name is Moira Furr, and I'm part of the Research in Biology program on the Durham campus. My project is titled Investigating the Neurotoxic Effects of a Heterogeneous and Environmentally Realistic Amalgamation of Micro and Nanoplastics. So, 460 million tons of plastic waste are produced every year, and when it breaks down, it's called micro and nanoplastics. Humans are exposed to micro nanoplastics daily via drinks, food, and clothes. They can infiltrate into the body's cell and tissue, causing harmful effects. They can also accumulate in the brain and cause things like neurodegeneration and interfere with neurotransmission. Microplastic research is an emerging field of study, and drawbacks of current MNP toxicity studies are that they do not use realistic conditions of what humans are actually exposed to daily. The most commonly studied microplastic is polystyrene and commercial polystyrene, which means that it is uniform in shape and size. Microplastic type, size, and shape, and even UV exposure can influence their toxicity, which means that realistic conditions are important to study because they could be potentially different from the single type microplastic. And in reality, humans are exposed to many different types of microplastics, not just uniform particles. So the goal of the study was to create an environmentally realistic microplastic mixture using 8 of the most common plastics humans are exposed to and to simulate photodegradation by UV. And to also determine if there is a difference in neurotoxicity of the heterogeneous environmentally realistic microplastics and the homogeneous microplastics that are commonly studied today through the analysis of locomotion behavior, dopaminergic neurons, cholinergic neurons, and neurotransmitter gene expression using the model organism C. elegans. I hypothesized that the environmentally realistic microplastics will show greater neurotoxic effects than homogeneous microplastics. C. elegans is a common model organism used for toxicity studies. They have a short life cycle, they are easy to maintain, and they are transparent, so that is useful for analyzing specific neurons and other things. So during SRIP, my main focus was to create the realistic mixture because no one had ever done this before. And there wasn't a standardized procedure, so I did a lot of research to figure out how I should go about going from a raw plastic item to um, microplastic particles. So to do this, I did a process of cutting and blending, and I filtered the mixture 2 times before evaporating um, the liquid off to leave me with a small layer of microplastic particles at the bottom of a beaker, which I then um, diluted to the concentration I wanted. So at the end of the study, the significance will indicate how using heterogeneous, environmentally realistic microplastics might provide more accurate and different results than traditional microplastic studies that only use commercial polystyrene or single-type microplastics. And research on this has never been done before, so it should be studied to most accurately represent the effects of microplastics on humans. Studies like this are needed because microplastics are continued to increase in production, and studies and information on their toxicity in our bodies are still widely unspecified, and including how we're exposed to them every day, and we still don't really know how they affect our bodies. Um, and the study also wants to raise concern about neurotoxic effects of microplastics and how they could impact our bodies and even lead to neurodegenerative disease. In the fall, I will do my analysis of dopamine in um, cholinergic neurons using GFP labeled C. elegans, and I will also do my analysis of neurotransmission genes. Through these, I'll find out how the environmentally relevant microplastics will affect specific neuronal groups and which neurotransmission genes are being affected. And these analyses will further indicate whether the heterogeneous microplastics are more neurotoxic than homogeneous single-type microplastics. For my project, I would like to thank Dr. Monahan and Mallory for their amazing mentorship and guidance, the NCSM Foundation, the Glaxo Foundation, and the Burroughs Wellcome Fund for funding, and the TAs in Rbio class of 26.